Hello, welcome back. Here in this mini lesson, we're going to go over animal viruses, which are the ones that affect us. We are technically animals, eukaryotic organisms, and so just like we had kind of life cycles or cycles of lytic and lysogenic bacteriophages, we're going to go through the different life cycles of replication for viruses based on their genome. So I'm going to walk us through kind of this handout, the biosynthesis of DNA and RNA animal viruses. But before we get to those actual steps, we can talk about how animal viruses actually get into our cells. And there's a couple different ways. Now, again, remember the virus we talked about being those two components, so the protein coat and the genome. Really, the most important part of virus is its genome, because it's the genome that needs to get into the host cell to make more virus. The capsid and the envelope and those structures are important for protecting the genome and for attaching it to the host cell, which is also very important. But really, when we talk about the virus, it just is that piece of nucleic acid, whether it's the DNA or the RNA. So the whole point of this attachment to the host cell is to eventually get that viral genome into the cell, because that's the only thing that it really wants to do, is to get into the host cell so then it can make more of itself. So in this first example, or this first method, is direct penetration. So direct penetration is there's going to be receptors on the cell surface, and they could be for anything. It could be for a hormone, or it could be for another, an antibody or something that the cell normally has on its surface. The virus that can infect that cell has something on its surface that when the two meet, it's kind of like a lock and key thing. So the virus finds the receptors recognize the virus. It's a trick though. It's not saying, oh, I recognize you, virus, come on in. The cell's saying, oh, hormone, okay, come on in, or whatever. So the virus tricks the host cell to let the viral genome get inside. So, excuse me, in direct penetration, the capsid stays out and just the viral genome comes into the host cell. In the next one, in membrane fusion, what we see is, just as the name implies, the enveloped virus. So direct penetration would be for non-enveloped viruses. Clearly, membrane fusion would be for enveloped viruses, where again, the receptor on the host cell is going to recognize something that's on those envelope surfaces, these glycoproteins. In the coronavirus example, they're called spike proteins. They bind to the receptor. It triggers basically a fusion of the membranes. So we can see the host cell membrane is fusing with the envelope membrane. And then all of those glycoproteins are left at the membrane. And then what goes into the cell is the virus with its capsid. But that's not going to do it any good. It can't do any of its replication if it's still stuck in the capsid. So something is going to trigger uncoding to just release that genome into the cell for it to start its replication process. The third example is called endocytosis. So it's very similar to the beginning of membrane fusion, where we get some connections between the host cell and the glycoprotein spikes on the envelope. But instead of just fusing the membranes, it triggers endocytosis, so a whole engulfing of the enveloped virus. This will eventually trigger fusion, like we see here, of the envelope and the vesicle, and then an uncoding, and eventually the viral genome comes in. I'm going to be posting some videos on dengue fever and HIV life cycles. They're really good animations of this method of entry into the cell. The dengue fever does it one way, the HIV does it another. So if you want to see some good animations of these entry mechanisms as well as the replication cycles, I encourage you to watch some of those videos I'll be posting. They're called HHMI Biointeractive, so Howard Hughes Medical Institute Biointeractive videos are the ones that will show some of these entry and exits and some of the replication processes that we're going to go through. All right, so let's get started. I've just made some kind of blank screen because normally what I would do in class, I would write all this stuff up on the whiteboard, but we don't have a whiteboard, so I'm going to pretend we have a whiteboard with my touch screen. So we're going to start with our DNA viruses. I'm going to kind of split this in half. So one half is going to be our double-stranded DNA, and the other half will be our example for single-stranded DNA. Before we get too far into these processes, I want to draw your attention to the very bottom of the handout. These things right here, these names of these enzymes and their abbreviations, because I'm going to be using those abbreviations in these drawings, so you're not lost. You'll know what I'm talking about when I say DDDP or DRDP, those types of things. So these are the enzymes that are responsible for the copying of the genetic material. So in our cells, when we're going through DNA replication, we call the molecule DNA polymerase. It was just DNA polymerase. And when we were in transcription, we just called it RNA polymerase, right? Does that sound familiar? So to be more specific, since we're going to kind of turn things a little bit wonky here with viruses, we're going to be more specific as to what those molecules, those polymerases are reading and what they produce. So for example, DNA-dependent DNA polymerase, DDDP, is what we would normally refer to as our DNA-dependent or DNA polymerase. So DNA-dependent means it is reading DNA. DNA polymerase means it's making new DNA. So a DDDP is an enzyme that takes DNA, copies DNA, and makes more DNA. That's what we do in our DNA replication. So that's nothing new to you. We're just calling it a new name. Compare that to DNA-dependent RNA polymerase. DNA-dependent, so we're reading DNA and we're copying it into RNA. This is what we would normally call our RNA polymerase, what we see in transcription. So it's nothing new to you. We're just calling it by a little bit more specific name. And the reason why we're doing this is because viruses sometimes will read RNA and make DNA, or read RNA and make RNA, or some kind of weird combination of those templates and their products. So use that handout to kind of help you follow along and track along which enzymes I'm using and describing for each one of these processes. Okay. Another thing I wanted to mention is, let's see, what else do I put up here? So the central dogma, DNA to RNA to protein. We should be comfortable with that concept. So genetic information is stored in the DNA. It is transcribed into RNA, right, mRNA, which is then used to translate that into protein. So you have to be comfortable with that understanding of that process. That was covered in general bio class. You can go back in some of those sections in chapter 7 if you need to review those DNA replication, transcription, translation concepts we skipped over in this class. We didn't test on those, but that's prior knowledge that's going to be really helpful for this. And then I'll leave this up here. And then another thing you need to know is mRNA. Well, I'm going to save that for another one because it doesn't really come into play on our DNA viruses. I'll talk about some mRNA rules when we get to RNA viruses. All right, so let's say we have a double-stranded DNA, and I'm going to use colors. That's kind of one of the nice things about doing this on a computer, is let's say we have a virus, and I'm just going to illustrate it with that capsid, and then a double strand of DNA. It's going to have, right, the complementary strands. So this is our double-stranded DNA. It's going to do the attachment, penetration, and encoding. So now what we have inside of our cell is we have the viral genome, right? This is viral double-stranded DNA. What it's going to do is it's going to hijack our cells, and then it is going to, via the process of transcription and DNA-dependent RNA polymerase, it is going to make mRNA, okay? So this is our viral mRNA. And then by the process of translation with host ribosomes and host amino acids, it's going to make, oops, let me get the right color here. It's going to make some protein chains, right? So I'm just representing your amino acid chain, which will eventually be your polypeptides to make your protein. So here's our process of transcription. It's a lot harder to write on the screen than it is on the whiteboard. And this is your process of translation. Okay, So at this point, we have our viral protein. That's the one part of our cell or the virus we need, right? We have the two parts. We need to make proteins and we need to make nucleic acid. We've made our proteins. Now we have to make our nucleic acid. So what's the template for when we make our DNA, right? Our DNA. So we are going to use host DNA-dependent DNA polymerase to make more DNA. Right? So we're just replicating the DNA. So these two things are going to come, oops, get the right color here. So these two things are going to self-assemble, running out of room, into more viruses, what we just started with, right? That's what we started with, a double-stranded DNA virus at the top. We used host enzymes and host processes to make more of the capsid protein and to make more of the enzyme. And so this is how it's going to make more. So that's going to be this first step up here. This is our double-stranded DNA. Herpes virus is an example of a double-stranded DNA molecule. If it's got an envelope, it will bud off. If it doesn't, it will just lyse out of the cell. Okay? So it's our double-stranded DNA virus. Okay? Now let's take a look. I think I'm going to change away from the orange though. Let's do green. That's a little bit of a better capsid protein. The orange is a little bit too light. All right, so let's start with what we have. We are going to now have a virus with a single strand DNA. It doesn't have a complementary strand. Just that's what it starts with. So that's what we're going to have to end up with. That's our goal. We have to make more of what we start with. We have to make more protein and we have to make more nucleic acid. The book proposes these two questions. How can you answer these two questions? Where do you get your mRNA to make your proteins? That's the protein part of the virus. And what's the template to make more virus? Genome. So that's the DNA replication or the RNA replication piece. All right, so once this single-stranded DNA attaches entries penetration into the cell, what we have is our single strand of viral DNA. Well, our cells can't recognize a single strand of DNA, so we use our own host DNA-dependent DNA polymerase to make the complementary strand. Okay, so this is still viral, so we use our enzymes, or it uses our enzymes to make the complementary strand. And then now that we have viral, double-stranded viral DNA, we just follow the same path that we did in the last process. So now we have viral mRNA, we do translation, we're going to get viral proteins. Right? So all of that is the same process using our host DNA-dependent RNA polymerase for transcription. Here's our translation, we get our proteins. And now for replication, it's a little bit trickier because we don't want to put in double-stranded DNA. That's not what we started with. So of our blue and our red strands, which one would be the template? Which one do you think we would use to make more of what we started with? Would we go red to make more red or blue to make more blue? What do you think? Think about that. Well, DNA replicates via complementary base pairing. So if we want to make more of the red strand, we would need the blue strand as the template. So that's what we would use. Okay, so we're going to use the blue strand with our host DNA-dependent DNA polymerase, just like we did to make the blue strand. We're going to use the blue strand as our template to make more red strand. And then we are going to self-assemble. And is this what we started with? It sure is. So DNA viruses are a little bit easier because they are most like what our cells normally do. So that central dogma, DNA to RNA to proteins, makes sense because that's exactly what they're tracking along. So that's going to be this one right here. So we've just did virus 2. So single-stranded DNA viruses, that would be like a parvovirus, okay? Using the host's enzymes and ribosomes to get its final product. Okay, so those are our DNA viruses. Next is our RNA viruses. So this is where I'll write up our central dogma again, right? So DNA to RNA to proteins. But I also wanted to point up here, kind of leave this up here, is when we talk about these pluses and minuses, these sense and nonsense strands of RNA, we need to know that mRNA is basically the same thing as single-stranded positive RNA. It is the positive, what we call the sense strand, that can be directly translated into proteins. So we'll divide this for our negative and positive. So when we as eukaryotic cells transcribe our DNA into a piece of mRNA, it is what we would call a single-stranded sense strand or positive strand of RNA. And when viruses have a single-stranded positive RNA genome, that genome can act just like mRNA. Okay, hopefully that will make sense as we go through. So now we're going to be working on the third one. That's this one right here. The coronavirus is an example of a single-stranded RNA positive virus. So imagine that if you wanted to. So we're going to start with our, so this is our single-stranded RNA positive virus. Okay, so I used purple in my last example as my mRNA because that was a product of transcription. So I'm going to try to stick with that same color scheme here. So if you are a single-stranded RNA positive virus, you got your capsid in green, you got your genome in purple, you're going to come into the cell and what do you have? That's your genome, okay? Viral mRNA. Now with mRNA, we've got to answer those two questions, right? How do we make protein? And how do we make more single-stranded RNA positive? Well, but there's no enzyme that reads RNA to make RNA. We don't have an RNA-dependent RNA polymerase in our cells. That's just not how our cells work. So one of the early genes that is transcribed, so I'll just kind of draw a little arrow over here. You know what? I'm going to put it on the other side. One of the early genes, or genes of what we call early transcription, is going to be a very special protein. I'm just going to make it a blue blob here. It's an enzyme, and this enzyme is RNA-dependent RNA polymerase, RDRP. So you can see that again down here. All the definitions are at the bottom of this sheet. RNA-dependent RNA polymerase. Now the virus has a way to replicate its genome. Okay? So it did a little bit of translation. So this was a kind of early translation to make this protein. But we can also do later translation into more of the capsid proteins. Okay, so that we're doing the rest of the translation like we would normally. So we've answered our one question. What serves as our, how do we get our mRNA? Well, the genome is the mRNA. It can come into the cell and just directly land on a ribosome and start pumping out proteins. Some of the proteins are going to be the capsid proteins. Some of the proteins are going to be this RNA-dependent RNA polymerase, because now we have to answer that second question. What serves as the template for more genome? Well, if you're going to go from sense strand to more sense strand, because of the complementary base pairing, you're going to need a negative strand in there somewhere. So I'm going to go like this. Using the host, this new viral RDRP that we just made, we are going to make, let me pick a color. I'll use dark blue. We are going to make the negative strand. So if this is my positive strand, right, that's our sense strand, we've now made a antisense strand, or the nonsense strand. So then again, we're going to use viral RNA-dependent RNA polymerase to complementary base pair more positive strand. See how that works? So that's why understanding your DNA replication is really important for understanding viral replication. So can we get more of what we started with? Well, we've got our capsid, we've got our genome, so I think we can. So here's our capsid proteins drawn very poorly, and here's our single-stranded mRNA, and that's exactly what came into the cell. So in this case, the single-stranded RNA positive could go directly onto a ribosome, start translating the enzyme needed to make its genome, and then also be translating the capsid proteins to make the capsid. And then with that newly translated RNA-dependent RNA polymerase, it can make the template, the negative strand, to then use to make more positive strand. Okay, it's very wordy, but hopefully you're kind of getting it. So that was our single-stranded RNA positive. So now we're going to take a look at single-stranded RNA negative. How's that going to work? Well, let's see. These weirdy viruses. All right, so if this is a negative strand, it's going to be that dark blue. So it's a negative RNA. So negative strand RNA cannot land directly on the ribosome. So when it gets into the cell, this is what we have, a negative RNA strand. Now, if this particular virus didn't carry something special in with it, it would just be done. There's nothing our cells can do with a negative RNA virus. It would just kind of sit there, probably would get recycled. But this virus carries its own enzyme. And this enzyme is called RNA-dependent RNA transcriptase, RDRT. Again, that's down here, right, in all of our descriptions. RNA-dependent RNA transcriptase. This enzyme has to come into the cell with the genome. So it's kind of hanging out here. Because before anything can happen, the virus has to use that enzyme to make the sensed strand, right? This is our positive. This was our negative. So it's using this enzyme, RNA-dependent RNA transcriptase, that came in with the virus to make the mRNA. Now what can we do? Well, we can do translation over here into our capsid proteins, and we can use again the enzyme. This enzyme again, the viral RNA-dependent RNA transcriptase that came in with the virus to make what? Our negative strand, right? The complementary strand to the positive, and that's what we need to make more of what we have. So can we make what we ended up with, or what we started with? We sure can. We've got capsid proteins, we've got our viral genome, and what else do we need to make? What else do we need to include in this finished product if we want successful viruses? Well, whatever we came in with. If we look at what we started with, we need to have one of our protein products be the RNA-dependent RNA transcriptase. That's what's going to allow the virus to replicate once it finds its next cell. All right, so those are our single-stranded RNA viruses, positive and negative strands. Okay, so we've covered that. So single-stranded RNA positive was coronavirus. Single-stranded RNA negative example is rhabdoviruses. Okay. Then the last slide is going to be some RNA viruses, which are double-stranded and retroviruses. So kind of split my screen, keep our central dogma up here, DNA to RNA to protein, and a reminder that mRNA equals single-stranded RNA positive, okay? So in these last couple viruses, we are going to have double-stranded RNA. All right, so let's see what we start with. We got our capsid, and we're gonna have our sense strand and our nonsense strand. All right, so that is our double-stranded RNA. So once that comes into the cell, it's going to look like this, just kind of for simplicity's sake. Right? It's not going to have any special enzyme with it because it comes in with a positive sense strand as half of its genome. So it can actually unwind. Right? So the positive strand, oops, let me get black, is translated because it can, right? mRNA equals single-stranded positive into that, oh gosh, what color did I think I made it this color. Is that right? This was our RNA-dependent RNA polymerase, right? So now it can make an enzyme that can copy and replicate its genome. We can also just get straight on translation into our capsid proteins and any other proteins or enzymes it needs. And now that we have our RNA-dependent RNA polymerase, we can use each one of those genomes as a template for more genome. So we can use this enzyme, taking the negative to make positive, and we can use that enzyme to make the positive be negative. So each one is being the template for the other one. So we have everything we need to make what we started with, right? So we've got translation of any positive sense strand we have. We have translation into our RNA-dependent RNA polymerase that allows us to now make our genome, which can get packaged in our virus. And that's what we started with. Okay, hopefully you're getting the hang of this pattern. Again, answering those two questions. How do we get our protein, right? This is basically how do we get our mRNA, which can be translated into protein, and what do we need to do to make more of our genome? Because remember, viruses are just those two parts. And then, all right, our last virus is going to be our retroviruses, which are two single-strand RNA positives. They are not complementary. They're two identical copies. I'm going to have to go small on this because these ones have a lot of steps to them. So we're going to have two positive strand RNA. But what we also need to incorporate, I've kind of run out of colors, maybe I'll do the light green, is we have a new enzyme. Okay, this enzyme, super special. We've heard it before back in our biochemistry or recombinant DNA technology. This is RNA-dependent DNA polymerase, also known as reverse transcriptase. Okay, so RNA-dependent DNA polymerase, just as the name implies, it reads RNA and makes DNA. So into the cell, once we have attachment and penetration, we get two copies of our sense strand. Now, for purposes of space and time, I'm just going to track one of these, but know that everything that I do will happen to both of these single-stranded RNAs. So, like our previous single-stranded RNA, it just went directly into translation to make the RNA-dependent RNA polymerase and make the capsid proteins. This does something different. It uses your RNA-dependent DNA polymerase to actually make our DNA, our double-stranded DNA, right? That's what we started with a couple slides ago, our red and our blue. So this is now viral double-stranded DNA. And what does it do with this double-stranded DNA? Well, it's going to incorporate it into a host chromosome. So you will have, if you get infected with a retrovirus, you will have parts of your chromosome will have viral pieces to it. And this infection is lifelong. So when you get infected with a retrovirus, HIV is the most well-known, I think, of retroviruses. That viral genome becomes a part of your chromosomes for the rest of your life. And it is in the chromosomes of your helper T white blood cells. So that's kind of the whole disease process of HIV and AIDS. But now we have the viral genome stuck inside this chromosome. Well, now what? How is it going to cause disease when it's stuck inside the chromosome? Well, just like our double-stranded DNA, when this virus is ready to present itself, it kind of lays dormant. It's called a latent infection or a dormant infection. It just kind of is hanging out in your chromosome, but eventually it will be expressed just like our double-stranded DNA gets expressed. So it's going to be translated into viral mRNA, which will be transcribed. I'm running out of room. I'm going all over the place. Into our viral proteins and our reverse transcriptase. And so now all of these things, I'm going backwards, gets into what we started with. Right? We've answered our questions. We've made our protein coat, we've made our genome, and we've made our reverse transcriptase that gets packaged in with this virus. And then that buds out of the white blood cell and then goes and infects more white blood cells. So this is how retroviruses replicate. They are two single-stranded RNA positives, but this unique property of this particular virus is it reverts itself into viral DNA, which then incorporates itself into the host chromosome, and then we call it a provirus. So this little area here is called a provirus. And then after a certain amount of time, the provirus gets expressed into the single-stranded RNA positive, which is then read as mRNA to make proteins, and then also gets incorporated as the genome into the virus. Whew, that's a big one. I should have given myself more room on that one, but that's this last one here. You can see how much stuff is going on with the retroviruses. All right, and again, some of the videos I post will be a life cycle of HIV, so you can track that if those animations might help. So for all of these drawings and all of the information that's on the handout, I want you to be able to express how animal viruses can enter the cell, how do they make copies of themselves, what tools do they use, answering those two questions. How do you make the mRNA to do the translation? How do you make more of the genome to get wrapped up in that capsid protein? All right. Okay, so that wraps up my drawing phase to the regular old PowerPoint. So once you have your virus assembled and ready to go, it can leave the cell either by bursting, just like the lytic cycle. If it's an unenveloped virus, it will just break the cell and be released. If it needs an envelope, it can bud. So this is showing budding off of the plasma membrane, but remember the viral envelope can come from the nuclear envelope, the Golgi body, the endoplasmic reticulum, or the plasma membrane. And the viral glycoproteins are some of those protein products that are made. I know I just showed the green circles as the capsid proteins, but they also have to make those surface proteins that are going to be on the outside of their envelope. So they do that, and they send those viral spikes up to the plasma membrane to wait for those mature viruses to come up and then bud out. And then it wraps itself in the host plasma membrane and these viral spikes that it made. Those animations that I was talking about show this actually pretty well. Okay, and then I think the last thing is just a slide showing persistent infections where you can get entry into the animal cell, synthesis and assembly, but instead of like a burst, like we saw with the burst time of bacteriophages, you might have a slow release of virions over long periods of time. So some viruses are released via these persistent infections, so it's more of a long-term chronic infection as opposed to a short, acute, couple-week kind of thing. All right, I know that was a really long video, but thank you for sticking with me. We have, let's see, I think we have a couple more videos in this particular group of culturing viruses and their role in cancer, and then we'll talk a little bit about viroids and prions at the end. All right, so I will see you next time. Bye.